Okay, so now, so it is 3 o'clock. So let's start today's open seminar. So I'm Okito Yamashita, team leader of the computational brain dynamics. Today, I'd like to chair this seminar. So first of all, thank you very much for participating in our seminar. Before starting the seminar, so I'd like just notify you the important things displayed in display. So four things. First, so reproduction is prohibited. And second, so reproducing all or any part of the content is prohibited without the author's permission. And third, this seminar will be recorded. So, and the video will be available on the Riken AIP website later. And fourth, so if you have any question or comments, so please scroll to the bottom of the page, bottom of the page, and click the Q&A tab. So you can put the question or comments in the box. So I think we accept both English question or Japanese question. So if you have a comment or question, please put in the tab. Okay, so good afternoon, everyone. So first of all, so thank you so much for attending today's seminar. So we are very grateful to have the opportunity to present our research activity in front of a many audience. So in our team, so we are developing AI technology for neuroimaging biomarker of the mental illness to realize precision medicine of the mental disorder. Today, we have 4 talks, including me. Each will take about 20 minutes talk and 10 minutes Q&A. We welcome any feedback from audience, as your questions or comments are very helpful to deepen our understanding. So if you have questions, so please use Q&A tab box. Okay, so now I start my talk. So mental disorder such as depression, schizophrenia, autism, and so on is becoming one of the biggest social problems. The number of patients increasing until 2030, so it will become the leading contributor to the loss of healthy years. However, the current diagnosis system, DSM-5, which is based on self-questionnaire and interview about their symptom, is not effective for treatment selection. For example, 60% of the depression patients are not cured with drug treatment. To increase the effectiveness of treatment, objective way to define mental disorder are anticipated based on the biological data such as blood, gene, and brain data. And now, biomarker studies of mental disorders are proceeding all over the world. Among various biomarkers, so resting state functional connectivity, which is an index of the brain network, is one of the promising candidates. Resting functional connectivity is obtained as follows. In the experiment, the brain activities during resting state measured for typically 5 to 10 minutes inside the MRI scanner. Subject just instructed to be relaxed without thinking any particular things. So this is a very easy task. So we don't need any demanding task, demanding cognitive task. That's why even patients can conduct this task. And this is a movie of the 10 minutes resting state MRI activity from 4 slices. And actually, there are about 100,000 time series data all over the brain. So these data are so complicated. Then in the analysis, the similarity between the distant brain regions are quantified with Pearson correlation of the time series data. So these lines are representing the region averaged time series data. And the red line and green line changing in the same way, while blue line and orange line changing the opposite way. In this case, we regard red region and green region are positively connected, while blue region, orange region connected are negatively connected. So in this way, we compute similarity between all the pair of brain regions, resulting in this kind of the connectivity matrix. And typically, this matrix contains the information more than 10,000 connections among the more than 100 regions. And although what the subject thinking is fluctuated by time, this temporal averaged functional connectivity is not random and actually known to be highly reproducible within subjects. And the previous study reported this functional connectivity is related to the anatomical connections divided into subnetworks related to perception, motion, and cognition. Also can be used to identify individuals like fingerprinting. And the brain network biomarker is constructed by applying machine learning approach to the functional connectivity matrices from many subjects. As example, let's consider the diagnosis biomarker, which discriminated patients from healthy control individual by individual. The brain network are represented by the summation of the connections. And unlike conventional biomarker consisting of the small number of the variables, brain network biomarker potentially has a large number of the connections. In this case, machine learning approach is very powerful too. So it automatically provides the optimal weighting parameters based on the exemplified samples. And this machine learning approach is very successful. And diagnosis biomarker of the depression, schizophrenia, and so on has already reported. Furthermore, more interestingly, using unsupervised learning approach, clustering approach. So stratification depression biomarker, which predicts effectiveness of RTSM treatment, so magnetic stimulation treatment, are also developed. And these results suggest the possibility to realize precision medicine based on the brain network biomarker. However, there is a big challenge to make the biomarker applicable to the public. And most of current studies use a small number of the controlled samples, typically 10 to 100 samples, within single hospital or single research institute. But real-world data is much more heterogeneous, contaminated by measurement bias due to the scanner difference or imaging protocol difference, or sampling bias, such as university hospitals tend to have more severe patients than local clinics. And developing the robust biomarker generalized to heterogeneous samples are now ongoing. From now on, so I'd like to explain two recent studies toward generalization to the real-world data. These studies are collaborated with SRPBS DECNEF project, in which ATR and Hospital University of Hiroshima, Tokyo, Kyoto, Showa participated. So first study is about the measurement bias and the harmonization, the measurement bias of the functional connectivity. So it is known, so fMRI image depends on the MRI scanner. So these are example fMRI image of one subject measured with two machines. One is named Berio and the other is named Skylar. Although both scanners have the same magnetic strength, measured with same imaging protocol. So you see image look very different. And actually, this causes a bias to the functional connectivity. And in this study, so we quantitatively investigated the measurement bias compared with the disease factors. Also propose a harmonization method which calibrates this measurement bias. So for that purpose, we collected and analyzed the traveling subject data sets consisting of the 400 samples and multi-site, multi-disorder data sets consisting of 800 samples. And the traveling subject data is very unique data set which enable quantify measurement bias. So 9 healthy subjects visited to the 12 sites in Japan to measure their functional connectivity repeatedly. Since subjects are same, but only scanners are different, so we can dissociate the measurement bias specifically using this data set. And the multi-site multi-disorder sets measured from 9 sites included in these 12 sites. And this data set provides disease factors, which is a main interest of this study. And to estimate the effect size of each factor, for each connection, we simply assume this. We assume simply this linear model consisting of the measurement bias factor and sampling factor and the disease factor and traveling subjects. We estimated the parameters of each effect size, parameter representing each effect size of each factors by fitting linear, regularized linear least square method. And this is a result variance contribution summarized over all 35,000 connections. And the disease factor is here. And actually, and the measurement bias is here. The measurement bias is larger than actually disease factor here, and indicating the importance of the reducing this bias. So our harmonization method is just ignoring this term after estimating this. So if we subtracted this measurement bias, so we could reduce this measurement bias by 30%. And it should be noted that this result is just a summary of the all connections. If we see individual connections, some connections have larger measurement bias, while others have larger disease factors. And this effect size map shows that, for example, this is a measurement bias. So larger measurement bias was observed in this bottom of the brain. For example, while MDD sampling factor, MDD disease factors are larger in the middle of the brain. And due to this kind of difference, so machine learning approach successfully extract important brain connections to some extent. Okay, the second study is on the depression biomarker, which generalized to the new data sets of the new sites. So based on the traveling subject harmonization method, and also cutting edge parcellation method called Grasser's parcellation. So we developed and evaluated the depression biomarker with a rather large number of samples, more than 1,000 samples from 9 sites. And we show the developed biomarker generalized to the new sites, which were not included in the discovery data, training data, with accuracy about 66%, with AUC equal 0.74. Data used for developing the biomarker is 700 samples from 4 sites, which measured in a SRPBS project, while data for validation is about 600 samples from 5 sites, which measured from another project. Since these projects are very independent, so imaging protocol as well as MRI scanners are different. And here is the performance of the developed biomarker. So the performance of the discovery dataset evaluated with nested cross-validation procedure and independent validation dataset is almost same, about 66% accuracy and AUC equal around 0.74. Neither overfitting nor underfitting was observed. And this is surprising, considering the number of the connections are much larger than the training sample size actually, and also heterogeneity of the data. And the bottom figure shows that 25 connections identified as statistically important. And 90 out of 25 connections, so MDD patients has weaker value than healthy control. And the largest difference is observed in left-right insular connections, this blue one. Also, some connections are found in depression, consistent with previous finding. And to our knowledge, this is the first depression biomarker discovered and validated with heterogeneous multi-site data set. Okay, so this is my final slide. So as I explained, so we are now on the way to robust biomarkers, which are generalized to real-world data. To advance more, we need to increase evidence level. We are here, so we need to increase this direction. So we need to develop and evaluate using more data from more sites. Currently, a couple of big data collection projects are advancing in the world. So we may access data size of more than 10,000 in the near future. On the other hand, so the performance of our depression biomarker based on functional connectivity may not be high enough. So to increase accuracy, so we need to explore new features which outperforms current resting functional connectivity. And the resting functional connectivity is temporal average statistics. But resting state activity has dynamically changing. So maybe using such richer information, so we may improve the performance. And today, so 3 members in our team will give the talk on exploration of dynamic feature of the resting state activity. So I hope so you enjoy their talk. Okay, thank you very much for your attention. So now, so let's move to the Q&A session. Okay, so we have two questions. Okay, so first question is, fMRI is a costly and bulky technology. What about the lowest cost and more mobile imaging technology? Yes, yeah, actually, fNIRS EEG biomarker study using fNIRS EEG study also ongoing, but I think they are more heterogeneous, and also information is not still enough to discriminate the individual level. But I think anyway, there are many attempts to make fNIRS EEG as a biomarker study using for biomarker study. And the second question is, would it be possible to use fMRI for therapy progress monitoring over short period? Yes, actually, there is a study from Hiroshima University. So they show the diagnosis biomarker, using diagnosis biomarker of brain network. Drug treatment changes the connectivity towards the healthy control. So, okay, so is there any other question or comments? Okay, so if not, so let's move to the second speaker. Okay, so Ikeda-san, are you ready? Okay, so second speaker is Dr. Shigeyuki Ikeda. So his talk title is Predicting Behavioral Traits Through Dynamic Modes in Resting State Brain Activity. So please start your talk.
Thank you for introduction. I'm Shigeyuki Ikeda. So it is an honor to be able to speak to you today. Today I'd like to talk about prediction of individual differences of human behaviors using dynamic mode decomposition. So this is collaborative research with structural learning team of Professor Kawahara in AIP. So let's move on to the background of these studies. So this study focuses on dynamic resting state functional connectivities. So as you can see, static resting state functional connectivities is computed from entire brain activities. So static resting state functional connectivities assume that functional connectivities between brain regions is temporarily stable. So on the other hand, previous studies reported that functional connectivities computed by moving time windows showed dynamical changes. So this is referred to as dynamic resting state functional connectivities. Previous recent studies reported that dynamic resting state functional connectivities explained more individual differences of human behavior than static resting state functional connectivities. The human behaviors are, for example, IQ, working memory, and so on. And in addition, dynamic resting state functional connectivity is effective in classification of psychiatric diseases. So there are two major analyses for dynamic resting state functional connectivities. One of the analyses is sliding window approach. The sliding window approach is a very classical method to evaluate temporal changes in functional connectivities using time windows. But there is no established way to choose optimal window lengths. The other analysis is hidden Markov models. So hidden Markov model is a relatively recent analysis to obtain brain state transition using Markov process. But the optimal number of hidden states is unclear. So dynamic mode decomposition has been used as an alternative method for dynamic resting state functional connectivities. So DMD can extract consistent brain activity patterns in both space and time. So using DMD, we can extract consistent spatial activity patterns which have coherent dynamics. So let me explain briefly the DMD here. So first, we prepared two data matrices like this. So each data matrix represents Fmri time series, and xt represent the spatial activity patterns at time t. So the n means the number of spatial dimensions. And capital X2 represent a data which was shifted one time point, like this. Here, we assume the existence of dynamical systems, capital A, like this. So in order to optimize capital A, in order to extract optimize capital A, we need to resolve this least squared problems. Consequently, we can get the dynamic mode by applying eigen decomposition to capital A. So eigenvector represent dynamic mode, and it is spatial activity patterns. So eigenvalue lambda represent temporal characterization of the corresponding dynamic mode, and argument lambda represent simple frequency of corresponding dynamic mode. So there is a previous study which applied DMD to resting state fMRI data. The previous study reported that temporal characterization of DMD is associated with behavioral and demographic measures. So on the other hand, there are no previous studies which investigate relation between spatial characterization of DMD and human behaviors. So our first question is that diverse human behavior can be predicted using dynamic mode. And our second question is that which frequency band dynamic mode are important in prediction. So based on two questions, we had 4 purposes. So our first purpose is to develop a method to predict human behavior using dynamic mode. And our second purpose is to investigate whether various behavioral measures can be predicted from dynamic mode. The third purpose is to investigate which frequency band dynamic mode are important for prediction. The last purpose is to investigate relations between spatial structure of dynamic mode and common and classical racing set networks. To achieve our purposes, we use publicly available human connectome project dataset. This dataset, anyone can use this dataset from this page. So this study included 829 young healthy adult subjects, and individual subjects completed 4 resting state fMRI scanning and 59 behavioral measures. So each round consists of 1,200 frames. The frames means the functional MRI image consisting of 900,000 voxels. The TR means the temporal resolution of the sampling in fMRI scanning. And 59 behavioral measures can cover 7 categories of human behaviors, such as cognition, in-scanner task, emotion, and so on. So each functional MRI image consisted of 900,000 voxels. So it is a huge spatial dimension data. To reduce spatial dimension of the resting state fMRI data, each functional image was parcellated into 268 nodes based on a previously defined functional template like this. So fMRI time series for each node was computed by averaging voxel time series within each node like this. So as a result, we obtained 268 fMRI time series. So this figure shows the overview of our analysis procedures. So as you can see, individual subject had 4 resting state fMRI data like this. So first, DMD was performed on individual rounds of individual subjects of resting state fMRI data like this. And then, as input features in prediction analysis, a gram matrix was computed using dynamic mode. So at that time, a gram matrix for each specific frequency band was computed using dynamic mode within each specific frequency band, like this. And then a mean gram matrix was computed by averaging 4 gram matrices, like this. And lastly, individual behaviors was predicted using Gaussian process regression and computed mean gram matrices, like this. So the number of dynamic mode is determined by the relationship between the number of spatial dimension and the number of temporal dimension of input data to DMD. So we obtained 268 dynamic mode by applying DMD to low resting state fMRI data. However, it is too few to capture the dynamics over the entire sampling time. So we need to increase the rank of capital X1 and capital X2 by making augmented data. To avoid these problems, we constructed a new augmented data matrix, capital X1, by stacking shifted fMRI data like this. So H represent the number of stacking data. So consequently, we stacked fMRI data 5 times because this is smallest value such that the number of spatial dimensions exceed that of temporal dimensions like this. So similarly, capital X augmented data matrix capital X2 can be computed by the same manner. So in order to investigate the significance of DMD, so we use temporal independent component analysis for comparison. So temporal ICA is similar to DMD, but temporal ICA cannot extract consistent spatial activity patterns which have coherent dynamics. So to perform temporal ICAs, we prepared data matrix capital X like this. Capital X represent fMRI time series and XT represent spatial activity patterns at time t. So temporal ICA divide capital X into capital A and capital S. So capital A represent mixing matrix in which each column represents spatial characterization, and capital S represent separating matrix in which each row represent temporal independent component. So this study used capital A to compute a gram matrix. So let's move on to the make a gram matrix. So this study made a gram matrix representing similarity between subject using dynamic mode. So however, it is difficult to compare dynamic mode between subject because there is no natural correspondence between the dynamic mode of different subjects. So we cannot compute the similarities between subjects using common similarity method such as cosine similarities. So to avoid this problem, we use Grassmann kernels. So let me explain briefly the Grassmann kernel here. So capital phi represent a set of dynamic mode of individual subject. So I and J represent subject index, and capital M represents the number of dynamic mode. So here, capital phi can construct a subspace. So, and the subspace can be represented as a point on the Grassmann manifold, like this. So two subspaces, similarity between two subspaces can be defined as a Frobenius norms between capital phi I and capital phi J, like this. So using Grassmann kernels, we computed a gram matrix for each gram fMRI data. So this study used 59 behavioral measures. So they can relate it to several subject demographics. They are age, gender, race, education, and mean head motion during resting state fMRI scanning. So these confounding factors were regressed out from individual behavioral measures. And we used the residuals as a target variable in predictions. So let's move on to the prediction procedures. So as prediction models, we used Gaussian process regressions, and the human connectome project data include the subjects which are members of the same family. So to avoid this bias and compute prediction accuracy, we employed leave-one family out cross validation. And then prediction accuracy was computed using Pearson's correlation coefficient and root mean squared error between true and predicted result. And the prediction accuracy was, p-values was computed using permutation testing. So and there were 59 behavioral measures, so we needed to collect multiple testing. So we employed false discovery rate collections like this. So this procedure was performed on DMD and temporal RCA separately. So let's move on to the prediction result. So this bar graph shows a comparison between DMD and temporal RCA. So asterisks shows the significant prediction result in both correlation and root mean squared error. So first of all, DMD outperformed temporal RCA like this. However, we did not observe significant prediction result in many behavioral measures like this. So let's move on to the prediction result in different frequency band. So entire frequency band was divided by intervals of 0.1 Hertz. So we obtained 7 specific frequency bands like this. And the top row represent the prediction result of using the entire frequency. So this result is the same as the prediction result of DMD shown at the previous slide. So as you can see, using using entire frequency showed better predictive power than using specific frequency band. And then 59 behavioral measures were classified into 7 categories. So as you can see, significant of the prediction result were observed. Most of the significant prediction results were observed in the category, the cognition categories like this. And on the other hand, low frequency of less than 0.2 Hertz seem to produce effective dynamic mode compared to high frequency of more than 0.2 Hertz like this. So let's move on to the investigation of the spatial structure of dynamic mode. So to extract representative of dynamic mode from entire all subject dynamic mode, so we employed modified k-means clustering. So dynamic mode consists of complex values. So the real parts and imaginary parts of DMD were stacked in individual row, individual run of individual subject like this. So and then the entire dynamic mode were aggregated like this. And then modified k-means algorithms was applied to the aggregate data. And in this clustering, the optimized the number of centroids was determined by elbow method. And then we computed amplitude and phase map in individual centroids like this. And lastly, the similarity between amplitude or phase maps and 10 common resting state networks was computed using cosine similarities. So these resting state networks have been known as well-established resting state networks in the neuroscience field. So let's move on to the spatial structures of dynamic mode. So this figure shows 10 centroids within 0 to 0.1 Hertz. So upper row represent the magnitude maps and the lower row represent phase map. So we found that several centroids showed similar spatial structure to resting state networks, but it is difficult to quantitatively interpret the similarities between the centroid and resting state networks. So next figure shows cosine similarities between the centroid and resting state networks in magnitude maps and in phase map. And then to quantitatively evaluate cosine similarities, these cosine similarities were thresholded with absolute value of 0.4. This is this figure shows the thresholded cosine similarities. So this threshold, the setting of thresholding is just empirical setting. So as you can see, most of the centroid showed high cosine similarity with several blessing set networks, such as medial visual areas, default mode networks, executive control. So default mode networks is the most famous blessing set networks. So our analysis revealed that blessing set fMRI data include default mode networks which have coherent dynamics. So on the other hand, in phase maps, the two centroids showed high cosine similarity with default mode networks like this. So let's move on to the spatial structure of dynamic mode within 0.1 to 0.2 Hz. So these figures show the centroid and cosine similarities. So as you can see, all centroids showed high cosine similarity with medial visual areas and executive controls. So from now, there are many results. So I'd like to explain briefly the other result. So this is 0.2 to 0.3 Hz. So as you can see, the one centroid showed high cosine similarity with executive controls. So this is 0.3 to 0.4 Hz, 0.4 to 0.5 Hz, 0.5 to 0.6 Hz. And this is last result, 0.6 to 0.7 Hz. So as you can see, the centroid of more than 0.2 Hz showed high cosine similarity, consistently high cosine similarities with executive controls like this. So this is the last slide. I'd like to summarize our result. So we had four purposes. So our first purpose is to develop a method to predict human behavior using dynamic mode. As a result, we succeeded in developing the prediction procedure with DMD. Our second purpose was to investigate whether various behavior measures can be predicted from dynamic mode. As a result, the prediction with DMD was successful, and the DMD outperformed temporal ICA in several measures. However, we did not observe significant prediction results in many behavior measures. Our third purpose was to investigate which frequency band dynamic mode are important for predictions. As a result, low frequency of less than 0.2 Hz may produce effective dynamic mode. Our last purpose is to investigate relation between dynamic mode and common racing set networks. As a result, low frequency of less than 0.2 Hz included dynamic mode related to some racing set networks, for example, default mode networks. And on the other hand, interestingly, the high frequency of more than 0.2 Hz included the dynamic mode related to the executive controls. Yeah, that's it. Thank you for listening.
Okay, thank you, Ikeda-san. So now let's move to the Q&A session. But before your question to your talk. So actually I have two questions on my talk. So I first answer their question, then you answer the question to your question. Okay. Okay. So first remained question is: do you have a way to analyze the brain without contact? I think it's almost impossible to actually measure the brain activity remotely. Precisely speaking, fMRI and MEG is not does not have any contact with head or brain, but their sensor should be very close to head. So my answer to this question is, so I think we don't have way to measure the brain activity without contact. And also we cannot analyze the data. Okay, and the second question is, there is research associating the gaze to the depression. The device has been used now. And I'm wondering, is your research related to gaze? Yeah, this is a very good question. And I don't think gaze is related to the functional connectivity in our study, because in our resting state experiment, a subject instructed to fixate his or her eye in the center of the screen. And I think gaze is not so main. Gaze may not affect the functional connectivity in our study. Okay, so now, so Ikeda-san, please answer the 3 questions. So first question to your talk. So first question is, why did you choose Grassmann kernel?
Yeah, as you said, there are many kind of Grassmann kernels. So actually, we did not try to other Grassmann kernels. So actually, and honestly speaking, there is no specific reason for selected this kind of Grassmann kernels. So if you have a chance, if I have a time, so we want to try to other Grassmann kernels.
Okay. So second question, so how should I use DMD and ICA differently?
Actually, I separately performed DMD and ICA on individual subject resting state fMRI data. So yeah, I separately performed these analyses.
And third question, so why does the dynamic mode depend so strongly on the frequency?
So this is aspect. This is the aspect of DMD. So DMD can extract spatial activity patterns which have a simple frequency. So so this is the reason for the dynamic mode depends so strongly.
Okay, and 2 additional questions for Thomas-san. And the first of his question is, so brain resting state might be fuzzy concept for patients who could keep intensively think or solve their mental problem during MRI scanning. How do you ensure or monitor the subject does rest? Would DMD be useful for monitoring the quality of the resting state? Wow, very difficult question.
So your question is a very important and important point. So actually, now there is no established way to confirm whether the subject will rest or not. So, for example, during the scanning, we checked the subject open their eyes or using the EEG. During the scanning, we checked the brain activities so the brain activity. So yeah, so it is difficult. Actually, it is difficult to check the subject whether rest or not. And then with DMD be used. Yeah, your second question is actually I cannot answer because I don't try it. I didn't try it. So yeah, I hope the DMD is useful for monitoring the quality of the resting state. And then
okay. So second question is how about the more controlled studies like stimulus-driven or evoked response, allowing more standardized comparison among different subjects?
Yeah, so there is other studies so which investigate whether the using the DMD features, they investigate whether the hand grasping can be classified using dynamic mode features. So yeah, and there have been already such studies investigate stimulus-driven or evoked response.
Okay. So if your second question about my depression study, so actually, yeah, there are studies which use tasks to discriminate depression patient. In particular, so depression patient has emotional function, dysfunction in emotional control, and some emotional stimulus is used to discriminate the healthy control and depression or subtyping of depression. Okay, so if there is no other questions, comments, so let's move on to the third speakers. So Ikeda-san, thank you very much. Okay. So third speaker is Dr. Yusuke Takeda. He will present titled Estimating Repetitive Spatial Temporal Patterns from Many Subject Resting State fMRI. So please start.
Thank you, Yamashita-san, for the introduction. I'm Yusuke Takeda, a researcher in Riken AIP and ATR. My talk is based on these two studies, so please refer to this paper for detailed information. First, let me start from brain activity during a task. Let's suppose that this square represents brain activity. Horizontal axis represents time, and vertical axis represents space, such as region, brain region. When we examine brain activity related to a task, we usually present the same kind of stimuli like this repeatedly. As a result, stereotypical spatiotemporal patterns appear after task onset. Here, spatiotemporal patterns are defined as activities represented by two-dimensional matrices of space and time, like this. Such patterns are formed by propagating activities. For example, when the activity propagated from brain region to yellow region, then from yellow region to red region, this propagating activity formed spatiotemporal pattern like this. So by extracting spatiotemporal patterns that appear after the task onset, we usually try to reveal signal flow related to the task. How about resting state? In the case of resting state, we don't present stimulus or observe response from subject. However, it is possible that spatial temporal patterns may appear after the onset of internal events, such as remember the past and imagine the future. Indeed, spatial temporal patterns in resting state brain activities have been widely observed in various brain regions and scale and species, such as mouse visual cortex, rat visual cortex, mouse whole brain, human electrocorticography. So, revealing spatial temporal patterns in resting state brain activities may unveil our internal brain activities, such as consciousness. However, there is a problem. In the case of resting state, we don't have stimulus or response onset. So the onset of spatial temporal patterns are also unknown. Recently, we overcame this difficulty with a method called spatial temporal pattern estimation, STEP for short. STEP estimates repetitive spatial temporal patterns from resting state brain activity data. Hereafter, I'm going to introduce this method, STEP. STEP assumes that resting state data contains several unknown spatial temporal patterns at unknown onset. In the case of this figure, resting state data contains 3 kinds of patterns, and each pattern appears several times. Here, the shape and onset of the patterns are unknown. This assumption can be mathematically formulated as follows. Resting state data Y consists of several spatial temporal patterns P convolved with onset time series U. Here, U takes 1 as the onset or 0 otherwise. The purpose of STEP is to estimate spatial temporal patterns P and their onset U only from resting state data Y. How to estimate P and U? STEP searches for the optimal P and U that minimize this objective function. This function represents the power of the residual error between observed and reconstructed resting state data. We solve this problem. We solve this optimization problem as follows. First, we initialize onset U by random numbers. By using this random onset, we estimate spatial temporal patterns P by the least squares method. Then we update onset U so that this residual error becomes smaller. By repeating this procedure, we can estimate P and U simultaneously. On the other hand, STEP has 2 hyperparameters, number and length of patterns. They are determined so that the reproducibility of estimated pattern becomes high. We divide the data into 2 parts. From each data, we estimate pattern, then evaluate the similarity between similarity of the pattern between the data, and the hyperparameters are selected so that this similarity becomes high. To verify the algorithm of step, we conducted a simulation test. We generated these 5 patterns, and they are assumed to appear at this onset. From these patterns and onset time series, we generated signal time series. In this time series, patterns are overlapping with each other, like this. By adding Gaussian white noise to the signal time series, we generated symmetric data like this. By applying step to this symmetric data, we estimated the spatial temporal pattern hidden in this data. These figures show the estimated pattern by step. The estimated pattern resembles the true ones. And these figures show the estimated pattern by 3 existing methods. It is obvious that this method failed to estimate the pattern. This is because the patterns are overlapping in the symmetric data. Anyway, step outperforms the existing method, indicating the validity of the step. By the way, currently many subject resting state data are available on the internet, such as from Autism Brain Imaging Data Exchange, Human Connectome Project, and UK Biobank. So we extended the step for big data, which we call big step, from many subject resting state data. It's data for subject 1, subject 2, and so on. Big step estimate common spatial temporal patterns across subjects, as well as the corresponding pattern in each subject, which we call subject-specific spatial temporal patterns. The main difference of the mathematical formulations between step and big step is here. This PSK is called as subject-specific spatial temporal pattern in big step. It consists of common spatial temporal pattern and its deviation from it. This deviation assumed to be small compared to the common spatial temporal pattern. From resting state data Y, big step estimate common pattern and subject-specific pattern and onset time series. The advantage of big step over step is the ability to handle multiple data separately. We don't need to make one big data by concatenating data across subjects. This ability reduces necessary memory to that of a single subject and reduce computation time needed for solving the optimization problem. We applied big steps to over 1,000 subject resting state fMRIs. The data were obtained from this Autism Brain Imaging Data Exchange, which includes subjects with autism spectrum disorder and typically developed subject. These figures show the estimated common spatial temporal patterns across subjects. 2 patterns with a length of 8 seconds were estimated. Horizontal axis represents time from 2 to 8 seconds. Vertical axis represent height to cut the brain from inferior to superior. These patterns transiently exhibit spatial patterns resembling those of well-known resting state network, such as positive activity of default mode network, negative activity of sensory motor network, and negative activity of visual network. The existence of the default mode, sensory motor, and visual networks suggests that these networks tend to appear in a time-locked way, like this. This is consistent with the previous report that default mode network activity is uncorrelated with the activity of task positive network. On the other hand, big step provides subject-specific patterns. For example, pattern 1 and 2 for subject 1, pattern 1, 2 for subject 2. So we compare the pattern between ASD and typically developed subject. This figure shows the differences in pattern 1. At 4 seconds in pattern 1 and here, large differences were observed as right prefrontal cortex and thalamus. On the other hand, at the same time in pattern 1, the default mode network exhibits the large positive activity like this. These results suggest that the differences are context dependent. That is, the differences in fMRI activities between ASDs and TDs do not always occur during the resting state, but tend to occur when the default mode network exhibits large positive activity, like this. Large difference occur when fMRI activity exhibit this active spatial pattern. This activity might relate to thinking about others. In summary, we developed a method to estimate spatial temporal patterns from resting state data, which we call STEP. Further, we extended STEP for big data, which we call big STEP. We applied big steps to over 1,000 subject resting state fMRIs, including ASDs and typically developed subject. This reveals the context-dependent differences of the fMRI activities between ASDs and TDs. Finally, we can test these two methods by using this MATLAB toolbox. These works were supported by the National Institute of Information and Communications Technology and the Japan Agency for Medical Research and Development. Thank you for your attention.
Thank you very much. So now let's move on to the Q&A session. So we have one question. So so first question, what is the machine learning method for abstracting from real waveform to the step? And what is its resolution? And what kind of information can we get from it? So there are 3 questions. So.
Machine learning, for the first question, this method is a kind of unsupervised learning. And this method use least squares method. Just use the least squares method. And for the second question, its resolution is depend on the kind of measurement. If we apply this method to fMRIs, its resolution becomes TR. In the case of this data from ABIDE, the resolution is about 2 seconds. And if we apply the method to MEG or EG, its resolution can be 1 millisecond. So its time resolution depends on the measurement, the kind of measurement. For the third question, what kind of information can we get from it? We can, this method extract yes, spatial temporal pattern that repeatedly appear in the data. So is it okay?
So I think so. In your study, you showed us, for example, group difference between ASD and so typically development
Yes.
subject, right? And also, if you apply your method to the maybe task data, you can find some task-related activity.
Yes.
So I think it highly depends on actually, depending on what's the data you apply, so you can extract some information. But what your method is doing is anyway to extract the spatial temporal pattern which appears repetitively appears in the data. Right.
Yes. Thank you for that.
Yeah.
Yes.
And how about applying image processing deep learning method to the 2D spatial temporal patterns? Have you tried, or the data is too noisy?
I have not tested this. I think yes, we can apply the deep learning methods to the 2D image obtained by our method. And yes, it may be interest. So to extract information from the pattern obtained by our method, applying this deep learning method may be interesting. Yes, I think thank you.
And additional question from Kobra-san. So since you observe the main difference between the patient and the healthy groups at a specific time point that coincide with high DMD activity, so do you think it would be sufficient to identify the effect merely with spatial filters? So do you really need a spatial temporal filter?
Spatials. Yeah. Yeah, I think yes. Yeah, I said that the main difference occur when the default model network exhibits large activity, but as this question said, I think spatial temporal filter is better. I think in this case, I think the large difference occur when default mode network have large activity followed by negative activity of visual network. So I think spatial temporal filter is better. Yes.
But have you ever actually tried to compare? Just my question. So have you ever compared just spatial pattern or spatial temporal pattern?
I have not checked, but this is very interesting suggestion. I want to try. Yes.
Okay. And okay, so one more question. So I propose a time series neural network, RNN. What do you think about it? So recurrent neural network, applicability of recurrent neural network to your purpose.
Yes. Currently, I have no clear idea, but I think this recurrent neural network, the collaboration of recurrent neural network and our method have good effect, I think.
Okay.
Okay.
So if no additional comment or question, okay, so now let's move on to the next speaker. So Takeda-san, thank you very much.
Thank you.
So next speaker is Dr. Hiroshi Morioka. So he will give us a talk titled Nonlinearity of fMRI reveals the primitive temporal structure linked to rest, task, and behavioral traits. So please start your talk.
Okay, thank you for the introduction. So in my today's talk, I will talk about our recent developments of the new analysis method for resting state fMRI data, which is a new type of the self-supervised deep learning. So here's this outline. So at first, I will talk about what is fMRI and why the analysis of the fMRI signals measured from the resting state brain is interesting and important. And I will also explain why actually it's difficult to analyze such data, resting state fMRI data, and then why little is known about the brain dynamics. Then after that, I will talk about our proposal of new nonlinear representation learning framework based on self-supervised deep learning. And then after that, I will show the result of the application of this method to the open resting state fMRI data. And then I will show the result of our findings, which is temporal primitives, which are primitive nonlinear temporal structures organizing non-stationary fMRI data. So my today's talk is based on our recent publication. So if you're interested in the detail of the method and the result, please read this paper. Okay, then let me start from the background. So fMRI is one of the most popular measurement modalities for the human brain. And by using fMRI, we can measure the blood oxygenation level happening in the brain, called Bolt signal. So this Bolt signal is supposed to be somehow related to the activity of the local neurons. So by measuring this Bolt signal, we can find which region is activated and which region is deactivated, and so on. This fMRI can measure the whole brain with the spatial resolution of 2 × 2 × 2 millimeter voxel, and the number of voxels in the whole brain is up to 100,000. And by using this fMRI, we can measure the brain activity as a time series manner, and the temporal resolution is up to 1 Hz. So the slow dynamics of this broad signal and this slow temporal resolution is one of the limitations of the fMRI, but still, because of this high spatial resolution, so this fMRI is one of the most popular methods for measuring the human brain activity. And the resting state fMRI means that during this fMRI scan, the subjects were just asked to keep awake and relaxed for 5 to 15 minutes. So a subject does not perform any explicit tasks during the scans. But actually, many studies found that even without any explicit tasks, we can say that the brain activities are not completely random, but actually we can find some level of commonality across subjects. This indicates that by analyzing this kind of resting state fMRI data, we can obtain some kind of ideas about the complex mechanism of the brain dynamics. And more recently, many studies show that, for example, we can predict the mental diseases of the subject, like depression or schizophrenia, or we can predict the behavior traits of the subject, like fluid intelligence level or something, by using this resting state data. So this is why the analysis of this resting state data is important. And usually, this fMRI signal is characterized by spatial co-activation patterns. For example, this is called functional connectivity matrix, and this is just a temporal correlation of the signals across regions. And as we can see, there is some kind of clustering structure in this data. And this means that some regions are frequently co-activated with each other. And by using, for example, clustering or RCA method, we can visualize those co-activation patterns like this. For example, we can find visual network or motor network and so on. And these co-activation patterns are very robust and consistent across subject and dataset. And those specific networks are also called resting state networks. And also many studies show that the transition from one co-activation patterns to other patterns are somehow probabilistic. And the difficulty of the analysis of the human signal is its high dimensionality and low SN ratio. So still there's no clear understanding about its mechanism. So in order to intuitively understand what is happening in the brain, kind of representation learning is crucial. But still, there are many challenges, like lack of labels or nonlinearity or non-stationarity of the brain. The first challenge is the lack of the label information. So as I explained, during the resting state measurement, subjects don't perform any explicit task. That means we cannot see what the subject was thinking about for its timing. So we cannot get the label information for its timing. So in this kind of situation, we have to use the unsupervised representation learning framework instead of the supervised learning. The representation learning means the kind of extraction of hidden representation of the data. And this representation is supposed to have low dimension and higher robustness and interpretability and higher generalization performance. And for example, we can use PCA, ICA, or HMM, or DSNA method and so on. And which method we should use depends on what we want to see or what we assume on the data. So still, there's no consensus about the best selection of the method. And the second challenge is the nonlinearity of the brain dynamics. So most of the conventional methods are based on linear assumptions like this PCA or ICA. So this is just a schematic image of those methods. And this is just a decomposition of the data metrics into a set of matrices representing time course and spatial patterns. But this kind of linear assumption makes no important aspect of the brain dynamics because it's supposed to be highly nonlinear. And one possibility is to consider such nonlinearity explicitly is to use the recently developed unsupervised learning framework, like variational autoencoder or GAN or recurrent neural network or something like that. But usually, so in the case of brain imaging, so it's kind of difficult to obtain enough number of data points to train such complex model. And also, those nonlinear models lack identifiability or interpretability of the result. Especially this interpretability is important because in the case of brain imaging, we want to know, we want to understand what is happening in the brain by using the learned representations. And the third challenge is the complex dynamics of the brain. So far, many studies try to characterize the brain dynamics by using some statistical method, like the switching behavior of the coactivation pattern by HMM, or extraction of propagation pattern, or switching behavior of connectivity by clustering method, or autoregressive model, and so on. But again, linear or stationary assumptions make no important aspect of the brain dynamics. So because of the challenges, still there's no clear understanding what is the fundamental factor organizing the dynamics and what is the best way to characterize it. So in order to tackle those challenges, in this study, we try to give new insight into the underlying dynamics of the brain by proposing a new nonlinear representation learning framework based on deep learning. Our proposal is a new type of self-supervised learning framework designed for spatial temporal patterns, and we call this method as space contorsive learning. And this method tries a feature extractor to find nonlinear temporal structures hidden in the data. And theoretically speaking, this method can be seen as a nonlinear version of traditional spatial independent component analysis, ICA. Okay, before explaining our method, so I should explain what is spatial ICA. So the spatial ICA is just a decomposition of the data metrics into a set of the matrices A and S, time course and spatial patterns. And this decomposition is based on the assumptions that the components are spatially independent of each other. And each component, each row, is characterized by a spatial pattern and a time course. Now this is the assumption of the linear spatial ICA. But our proposal is a nonlinear version of this. So that means we assume that the data metrics were generated from a nonlinear mixture of the source components. So these source components are unknown, and the F is also unknown. And the advantage of this nonlinear version of spatial ICA is that we can consider wider definition of the temporal structures to characterize the components. For example, we can consider nonlinear AR model or something. But usually this kind of nonlinear ICA lacks identifiability. So there's no guarantee that we can estimate those source components from the data. But recently, we found that we can actually guarantee the identifiability by assuming the specific structure of the data. And our proposal here is extension of this method. So our proposal is a self-supervised learning framework based on spatial function non-stationarity of fMRI signals. And we call this method as space contorsive learning. So self-supervised learning is recently getting attention in deep learning field. So self-supervised means that so we train the feature extractor in supervised manner, but we don't use specific labels like cat or dog. But instead, we use the kind of labels inherent in the data. For example, in this study, they proposed to use the information of the relative locations of the two patches. For example, first patch was selected from the center of the face of this cat, and the second patch was selected from this left ear. Then the classifier has to predict that the location of this second patch is 3. And in this study, they proposed to use the augmentation of the data, like cropping or flipping. And by comparing two patches, the classifier tries to predict those two patches were obtained from the same original image or not. So as we can see, even though we don't use the specific labels like cat or dog, we can still train the feature extractor to represent the data pretty well. So this kind of method can be used for the representation learning of the data or pre-training of the supervised learning if the number of the label data is not enough. And our framework, space contrast learning, is one of the such method, self-supervised learning framework. So again, this figure shows the assumption of the generative model. So we assume that these observed signals are generated from a nonlinear mixture of source components. Those source components and this nonlinear mixture is unknown. And this is the ordinary assumption of the nonlinear spatial share. But we additionally assume that we can divide, we assume the spatial non-stationarity of the source components, which means we can divide the brain regions into smaller parcels from 1 to k. And each of the parcel, we assume that the distribution is stationary. And this kind of assumption is reasonable in the case of brain imaging because there's many evidence that the brain functions are somehow localized to each region. And based on those assumptions, we train a nonlinear feature extractor and multinomial logistic regression classifier. This feature extractor takes a single time series as an input, then output a set of feature values. Then this classifier tries to predict where that input came from the parcel level 1 to parcel level k. So based on that information, the difference between actual level and the prediction, we update this classifier and this feature extractor by backpropagation manner. So yeah, definitely this is a self-supervised learning framework because we don't use the label, but we use the artificial label, which can be obtained from the data itself. And after training, the output of this feature extractor, the feature values are supposed to represent these original source components. So this is the idea of our method. And for the feature extractor here, we used deep convolutional neural network, which takes a single time series as an input and output a set of feature values. And the important point here is that, so in order to realize this classification, this feature extractor has to decompose this time series into distinctive components somehow. So otherwise, this classifier cannot predict the parcel level where the input came from. And in order to do that, this feature extractor has to find the component-specific temporal structures hidden in this time series. So that is the most important job of this feature extractor. And we applied this method to the Human Connectome Project resting state fMRI dataset obtained from over 1,000 subjects. This is one of the biggest open dataset available on internet. Okay, then let me show the result. So at first, we found that the feature extractor was trying to extract the specific temporal structures specific to each component. So this is component 1, component 2, 3, and 4. So yeah, apparently those temporal structures are very distinctive across components. And we found that those temporal structures appeared frequently and consistently across the whole dataset. And we call those temporal structures as temporal primitives because we are thinking that those temporal structures are the primitive structure composing the resting state fMRI. And we also found that the temporal patterns of those temporal primitives were not always the same, but actually those temporal patterns were hugely modulated for each event. This is the visualization of the modulations. For example, sometimes the frequency of the temporal patterns was different. Sometimes we see additional peak at some point, and sometimes we see different lengths of plateau and so on. So this result is kind of evidence of the nonlinearity of the brain dynamics, and this result indicates why we have to use nonlinear feature extractor to find those temporal primitives. And also we found that those temporal primitives were organizing the spatial co-activation patterns like this. Especially this component 2 showed a wide variety of co-activation patterns, and many of them are actually pretty similar to the well-known resting state networks. And the other components also showed very interesting co-activation patterns. So, this is kind of evidence that those temporal primitives were the fundamental factors organizing the co-activation patterns during resting state. And lastly, we checked the relationship between the activity of those temporal primitives to the behavior traits of the subject. For example, the activity of this component 1 has the higher correlation to the working memory skill of the subject, and the activities of the C2 and C3 had the relationship to the fluid intelligence of the subject. So this result indicates that those temporal structures were not simply abstract or something, but actually those components have important physiological meaning in the brain dynamics. Okay, then let me summarize. So the motivation of the study is to understand the underlying nonlinear dynamics of the brain during resting state. And then we propose a new method for the space-contextual learning, which is a new nonlinear spatial framework based on deep learning. And by applying this method to the open dataset, we found the primitive temporal structures composing the resting state fMRI data. And those temporal primitives were organizing the time-varying spatial co-activation patterns and also the relationship to the behavior traits of the subject, like working memory skill or intelligence level also. And again, so my talk is based on our recent publication. If you are interested in more detail, please read this paper. And the possible future direction is like this. So actually recently we are working on the application of this method to the biomarker of mental diseases. And we are also interested in application to other imaging modalities, including some type of biomedical imaging data. And we are also interested in finding of fundamental brain dynamics, which can explain micro to macro phenomena in a unified manner. And we are also trying to propose a new algorithm to better understand the brain dynamics. And even though I'm not sure how to do this, but possibly we want to use our finding from the brain to the neural-inspired AI system. And if you're interested in possible collaborations, please contact us. Okay, that's it. Thanks so much.
Okay, thank you very much. So now let's move on to the Q&A session.
Yes.
Okay, so we have several questions. Okay, first, so can we use this method to analyze changes in emotion and affect? But I think this question is about Takeda-san's method.
Okay.
Because this question is coming very early of your talk. So I think Takeda-san, can you answer this question?
Yeah, I hope we can, but at this time I don't have any evidence that our method can do it, but I hope it.
Okay. So next question is, have you considered combining the result of EEG and fMRI?
Okay, I see. So yeah, actually, that's an interesting direction, I think. So yeah, in this study, we applied our method only to the fMRI data and didn't apply to other imaging modalities or the combination of other modalities. But actually, the combination of EEG and fMRI is very interesting. So both have the a specific advantage. fMRI has the advantage of spatial resolution, but the EEG has the advantage of temporal resolution. So possibly by applying our method to the combination of EEG and fMRI, we can find some kind of the common components which can explain the signals of both of EEG and fMRI. So I think that is interesting direction, but so far we didn't do it. Thanks so much.
Okay, so next question. So is it possible to analyze and visualize the connectomes more accurately by using latent feature of VAE? I think this question is maybe related to the your presentation, your previous.
This
Yeah,
one?
this one, I think, related to this Kim's et al. Studies.
I see. Connectome. Connectome means the connectivity patterns, maybe. So actually, to be honest, I'm not pretty sure. So what kind of data they used for extracting the latent variables? But I suppose their input is the probably spatial patterns or spatial temporal pattern of the fMRI signal. And I think they show that actually by using the variational autoencoder to the fMRI signal, they can clean up the signals compared to the original data. I mean, they can increase the SN ratio by using variational autoencoder. But I'm not sure we can input connectome to this variational autoencoder model. So I'm not sure either there's such study or not. Yeah, possibly we can improve the quality of the connectome by using VAE, but I'm not sure.
So I think so their study used the image as input and the image as output, right? And
Yes.
you say that anyway, their output is denoised version of the input.
Right.
In some sense.
Yes,
So
yes.
then maybe it is useful to denoising the image is maybe useful for completely clear connectome pattern.
Yep.
I think they haven't tried.
Yeah,
right?
I agree with that. Yeah, connectome can be considered as an image data in some sense. So probably.
My experience of the VAE to the fMRI data, so actually VAE cannot reconstruct the original image very well. It's very smooth version of the input image. So I'm not sure whether, VAE output is enough, contain enough information of the original one. So that's another aspect. Okay, so this one is, okay, so do you use actual medical data to validate the learning accuracy?
Actually, we didn't use medical data. I mean, I used only the healthy subject data, and all of the subjects were somehow healthy. And the data set didn't include the subject which have the mental disorders. So yeah, actually, we are now working on the application of this method to the biomarker studies. So which means, so we are trying to predict the mental disorders like depression or something by using our method. But so far, we haven't checked the classification accuracy between the healthy subject and those patients.
Okay, so 2 more questions. So what are time 0 second locations in your temporal structure resulting from your method? Just min-max peak locations?
Sorry, I didn't explain detail about that. So
it's very
yeah. Okay, so I selected time point 0 as the peak of the signal, the maximum absolute value of this black line. So there's no special meaning of this time point 0. You know, during resting state, there's no external stimulus, or there's no specific timing of the task of the subject. So we cannot decide which timing is the 0 actually. So those temporal patterns were extracted from the how to say, the very random timing and locations for the whole data set. So we just sorted the extracted time series into the based on the cross correlation, and then decided the peak timing as 0. So there's no special meaning out of this location 0.
Okay. So last question is, so is neuro-inspired AI mean STDP?
STDP. What is that?
Spike timing dependent plasticity.
I see. Okay. Yeah, possibly something like that. Yeah, you know, STDP is, I'm not sure. STDP is also possibly being used for the training of the deep learning or something. But so yeah, to be honest, I don't have a clear image what is neuro-inspired AI and how to realize that. So yeah, possibly by finding some kind of interesting phenomena happening in the brain, like by finding how the connectivity is organized or something. So we can possibly propose a new type of the neuro-inspired AI system, which can deal with the analysis of the big data in more, how to say, complex way or something. But so far, I don't have any specific ideas how to realize neuro-inspired AI and what it means actually. I'm sorry about that.
I think fMRI signal, it is very difficult to relate fMRI signal to STDP, in my understanding. So maybe, yeah. So yeah, it's not so direct.
I see.
Related to the finding in fMRI study to the STDP. So maybe another aspect.
I see. Yeah, that's true. Yeah, so far we applied only to the fMRI data, but if we apply to the other modalities, like calcium imaging or spiking data, possibly we can find more detailed patterns. So maybe that can be helpful for the future development of that kind of systems.
Okay, so now last question. I think those temporal structure fixed in time or in amplitude? So I think
Okay. So the amplitude can be different across subject. So actually the HMI signal is pretty noisy. So sometimes those patterns are pretty difficult to find because of the noise. And sometimes the amplitude of the signal is pretty small and compared to the noise. And that actually makes it difficult to find those patterns. And regarding the length of the temporal patterns, so actually the temporal length actually can be defined for each subject. So yeah, to be honest, I didn't check the difference of the length across subject, but as far as I can see from this plot obtained from the whole subject data, for example, sometimes the temporal patterns are slightly shorter. I mean, the frequency is higher compared to other timesheets. So this is kind of evidence that the length of the timesheets can be different a little bit across subject or across the timings.
Yeah, very interesting. Okay, so now let's close today's seminar. So before closing, let me advertise the coming AIP Open Seminar schedule. So Morioka-kun, can you off?
Oh, sorry.
Okay. So this is the coming AIP Open Seminar schedule. So from next week, so we have a very interesting series of talk from language information access technology team and continuous optimization team and so on. So if you have an interest, please join this talk again. Okay, so let's close up today's seminar. So thank you for your participation and also thank you for speaker for your presentation. Okay, let's close.